Hello everyone. Today, we are going to discuss and explore beta-lactam antibiotics. Let's get started. Question 1. What is the mechanism of beta-lactam antibiotics? Correct answer, 3. Inhibition of bacterial cell wall synthesis. Beta-lactam antibiotics inhibit the transpeptidase enzyme, also known as penicillin-binding proteins, PBPs, which is crucial for cross-linking peptidoglycan chains in the bacterial cell wall. This results in weakened cell walls and ultimately leads to bacterial lysis. Let's look about the other options. Inhibition of protein synthesis. This mechanism is seen in antibiotics like macrolides, aminoglycosides, and tetracyclines. Inhibition of DNA gyrase. The mechanism of action for fluoroquinolones. Inhibition of folic acid synthesis, seen in sulfonamides and trimethoprim. Question 2. Which of the following antibiotics is not a beta-lactam? Correct answer, C. Vancomycin. Vancomycin is a glycopeptide antibiotic that inhibits bacterial cell wall synthesis by binding directly to peptidoglycan precursors, not by targeting penicillin-binding proteins. All other options contain the characteristic beta-lactam ring and are classified as beta-lactams. Question 3. Which of the following is a beta-lactamase inhibitor commonly combined with beta-lactam antibiotics? Correct answer, A. Clavulanic acid. Clavulanic acid inhibits beta-lactamase enzymes, which are produced by certain bacteria to degrade beta-lactam antibiotics. This combination helps extend the antibiotic spectrum against beta-lactamase-producing bacteria. Let's look about the other options. Azithromycin, a macrolide antibiotic. Metronidazole, effective against anaerobic bacteria but not a beta-lactamase inhibitor. Ciprofloxacin, a fluoroquinolone antibiotic. Question 4. What is the role of probenecid when co-administered with penicillin? Correct answer, B. Inhibits the renal excretion of penicillin. Probenecid competes for renal tubular secretion, slowing the excretion of penicillin. This increases its concentration and prolongs its activity in the body. Question 5. Which beta-lactam antibiotic is effective against Pseudomonas aeruginosa? Correct answer, B. Piperacillin-tazobactam. Piperacillin-tazobactam has extended-spectrum activity against gram-negative bacteria, including Pseudomonas aeruginosa. Let's look about the other options. Amoxicillin, limited activity against Pseudomonas. Benzylpenicillin, ineffective against Pseudomonas. Ampicillin, not active against Pseudomonas. Question 6. The primary reason for combining amoxicillin with clavulanic acid is? Correct answer, A. To extend the spectrum against beta-lactamase-producing bacteria. Clavulanic acid neutralizes beta-lactamase enzymes, allowing amoxicillin to remain effective against resistant bacteria. Question 7. Which of the following is a common mechanism of resistance to beta-lactam antibiotics? Correct answer, C. Mutation in penicillin-binding proteins. Mutations in penicillin-binding proteins reduce the binding affinity of beta-lactam antibiotics, leading to resistance. Let's look about the other options. Efflux pump overexpression, more common in tetracycline and fluoroquinolone resistance. Alteration of ribosomal binding sites, related to macrolides and aminoglycosides. Production of reactive oxygen species, not a mechanism of resistance. Question 8. Why are beta-lactam antibiotics generally less toxic to humans? Correct answer, C. Humans lack penicillin-binding proteins. Beta-lactam antibiotics specifically target PBPs, which are absent in human cells. This selectivity minimizes toxicity. Let's look about the other options. They target bacterial protein synthesis. This is incorrect, because beta-lactams target cell walls. They inhibit human cell wall synthesis. Human cells lack cell walls, so this doesn't apply. They are rapidly metabolized in human cells. Beta-lactam toxicity is not related to metabolism. Question 9. What is the main adverse effect associated with penicillin? Correct answer, C. Hypersensitivity reactions. Hypersensitivity reactions, such as rashes, fever, or anaphylaxis, are the most common adverse effects of penicillin. Let's look about the other options. Nephrotoxicity, more common with aminoglycosides. Hepatotoxicity, rare with penicillin but seen with drugs like isoniazid. Ototoxicity, typically seen with aminoglycosides. Question 10. Beta-lactam antibiotics are less effective against atypical bacteria because? Correct answer. A. Atypical bacteria lack a cell wall. Atypical bacteria, such as Mycoplasma, lack a cell wall, making beta-lactam antibiotics ineffective as these drugs target cell wall synthesis. Let's look about the other options. Atypical bacteria produce more beta-lactamase. While true for some bacteria, this is not the primary reason beta-lactams are ineffective against atypical bacteria. Beta-lactam antibiotics cannot cross the outer membrane. This applies to gram-negative bacteria, not atypical bacteria. Atypical bacteria actively pump out beta-lactams. Efflux pumps are a resistance mechanism, but not the main reason for beta-lactam ineffectiveness against atypical bacteria. This quiz has covered key aspects of beta-lactam antibiotics, including their mechanism of action, spectrum, resistance, and clinical uses. Thank you. Please subscribe our channel to watch more videos on tutorials.